DNA replication, transcription, and translation. In this video, we're really going to get into the main idea of genetics and how our genes actually give us our appearance and how they actually form into making enzymes and everything that actually makes us look like who we are and allows us to function normally and effectively. So let's get into it. DNA replication. So first we're going to talk about DNA replication. This process occurs when the DNA needs to be produced for a new cell. Let's first take a look at the DNA strand itself. When the DNA strand is replicated, first the two strands of the DNA are separated by a unit called DNA helicase. This helicase basically just breaks hydrogen bonds that form between the bases or the nitrogenous bases of the DNA. Next, a unit called DNA primer comes in, and this connects the DNA strands and acts as a binding site for DNA polymerase unit to attach to. The DNA polymerase unit basically creates complementary base pairs of the two strands. So this is creating another strand based on the template that it has of the original strand. And this forms two DNA helixes. But there's one catch to DNA polymerase. It can only create complementary base pairs in the 3' 5' direction. So again, remember, DNA strands have two directions, 3' and 5'. This DNA polymerase can only go from 3' to 5'. But one of the strands runs in the 5', 3' direction, as again, DNA runs anti-parallelly. So in order to replicate this anti-parallel strand, the DNA polymerase makes use of many primers and completes the strand in fragments. The strand that runs in the 3' 5' direction will complete it without the use of fragments and is called the leading strand. And the strand that makes the use of the fragments is called the lagging strand. The fragments which the lagging strand is completed by is called Okazaki fragments. So, again, the lagging strand is completed in Okazaki fragments and uses many primers, while the leading strand can just be done in one go. Finally, the two strands that are formed are binded and proofread by a unit called DNA ligase, which basically just secures the two new strands that are being made in the lagging strand and the leading strand, and it makes sure that everything is going correctly, there are no mutations. But the DNA ligase can't completely work 100% of the time to remove the mutations, and this is why we sometimes see disorders. Anyway, the DNA ligase sticks the strands together, and again, make sure that they are correctly paired. And this makes up DNA replication, as there are now two strands of DNA where there used to be only one. Now, when DNA replicates, it runs into another problem. Each DNA has, again, a 3' and 5' end, as we know, and these ends are denoted because of specific sequences they have on those ends. But when there's DNA replication, when the 5' end is trying to find a complementary, when the sequence of the 5' end is trying to produce a complementary strand for the other strand, then a 3' end can't be added to that 5' end of DNA. So basically, that section is just ignored, and the actual DNA sequence is just becoming shorter and shorter and shorter, since the ends are just kind of being ignored each time. But in order to prevent this, there is a special sequence on the DNA called telomeres. These do not contain nucleotides that code for genes, but instead they have a short repeated sequence. This sequence protects the DNA from becoming shorter and shorter, and it basically allows it to become, it basically prolongs this shortening, and it allows it to stay longer for a longer period of time. It acts as a buffer zone, so it doesn't completely stop the shortening of the DNA, but it just, again, acts as a buffer zone and prolongs this shortening from happening at an increased pace until the DNA is no longer there. So another function of the telomere is to prevent the ends of the DNA from activating systems of monitoring DNA damage. So the ends of the DNA release transduction signal pathways, which create a signal that cause the cells to stop dividing. So the telomeres basically block the ends of the DNA so that these signals cannot be sent, and so the cell allows more reproduction and it can still continue to thrive. So again, there's two functions of telomeres. One is it prolongs the shortening of the DNA, and two, it stops the cell from being unable to divide. Packaging. Now, when packaging of DNA. So DNA in a eukaryotic cell, or a cell that has a nucleus, is packaged in a very accurate and efficient way. Because if it weren't for the precision of the DNA packaging, there would be no homologous chromosomes, because the two chromosomes that were supposed to be homologous would not have the same genes in the exact same places, and they wouldn't be able to match up. So how is DNA packaging able to carry out such precision in this chromosome? Or how is this DNA even packaged into the chromosome? Well, there are proteins called histones that help this DNA become compact and organized. So histones are positively charged, and they allow the negatively charged DNA to attract to it, since opposites attract, positive and negative. These histones form bundles of 8, and the DNA wraps around the bundle 2 times. These bundles and DNA together form a structure that represents kind of like beads on a chain. The beads in this instance are called nucleosomes, and the whole structure is called linker DNA. So the beads and on the chain are called linker DNA. Each nucleosome, or the beads, has a diameter of 10 nm. When the DNA is trying to be transcribed, the histone leaves the linker DNA. So again, the beads leave the linker DNA, and there's no nucleosomes. And this allows the DNA to be transcribed because it's not looped around the histones. So after the linker DNA is formed, this still has to go through quite a bit in order to become a full chromosome. So the next level of packaging is the coiling of this linker DNA. On each of the chromosomes are H1 histones, a special type of histone. And this allows the linker DNA to coil up tightly, creating a fiber that is 30 nm in diameter. Next, loops are formed by these fibers, almost looking like a flower, and these form a fiber that is 300 nm. So slowly this fiber is becoming bigger, but also more condensed, as we see looping of the flower, we see coiling, and after this fiber is compressed and forms a 250 nm fiber. Again, this compression is able to happen because of the certain histones called H1. This creates the chromatid of a chromosome. Again, this process is highly precise, and it is thus able to compress the large DNA strand into a tiny, tiny package of a chromatid. So just now I mentioned the transcription of DNA, and now I'm going to go in a little bit of detail into that. So when we think of DNA, what is the purpose of it? Obviously, it is to store genetic information, to store all the information that makes us, us. But how does this information convert to become us? Is it not just the mere fact that they are in the DNA that shows that our hair is brown or black? What is making these instructions or DNA and implementing it or creating our characteristics? This is where the process of transcription and translation comes in. These processes together take the instructions that are in the DNA, and they eventually convert these into proteins, which create enzymes that create pigment for our hair or create different parts of our body based on the genes. So first is transcription. When we were talking about DNA replication, we mentioned how the DNA polymerase creates a complementary strand by using the leading or lagging strand as a template. Well, transcription basically works the same way, but instead it only uses the leading strand, and it does not create a complementary DNA strand, but creates a complementary RNA strand. RNA sounds a lot like DNA, but is a bit different. First of all, DNA is double-stranded, but RNA is single-stranded. Also, DNA carries information for genes of organisms, but RNA holds the information regarding instructions for creating the proteins based on the genes. And also, DNA has the bases thymine, adenine, guanine, and cytosine, whereas RNA has the bases adenine, guanine, cytosine, and uracil, where uracil basically takes the place of thymine and it pairs with adenine. So transcription is when the complementary strand of DNA is taken as RNA, and this RNA is created not by DNA polymerase, but by RNA polymerase. So this strand then sets the path for the genes turning into proteins, which can then be expressed. So this RNA has the complementary information. It is called mRNA. But the problem with this RNA is that it contains some extra information that is not needed for the proteins. And it's not needed for expressing the genes. They're just there because it was there in the DNA, and this was then reflected or transcribed into the RNA, but it's really not needed for the proteins. It's just needed for the genetic code. So these extra pieces of RNA, called introns, are basically just sliced out, and the actual parts that we need for gene expression, or exons, are kept. So the introns are cut out of the mRNA, and the exons are all put together, forming the mRNA strand. Also, on the 3' end of the strand, a sequence is added called the poly A tail, which is basically a sequence of 50 to 250 adenine nucleotides, and then on the 5' end there is a 5 cap segment, and this is again just a sequence of guanine nucleotides that are added onto the strand. These caps protect the RNA from all the things in the cell that may try to break it down. So this RNA also contains regions called UTRs. These UTR regions found on the mRNA are found right before one of the caps or poly A tails, and these are untranslated portions used for ribosome binding. And we'll discuss this in the next important step of gene expression. And the UTRs are not technically exons because they don't actually code for gene expression, but they are kept into the mRNA and they're not spliced out because they are needed for the next step in this process. So after all this modification has been done to the mRNA with the splicing out the introns and all the caps put on to each side, the mRNA is now ready to exit the nucleus. So it will exit the nucleus so that it can attach to a ribosome, which will use the instructions that the mRNA has in order to create the actual proteins. The ribosome is another organelle of the cell, like the nucleus, and its job is just creating proteins. So anyway, the mRNA will travel to the ribosome, and it will be inserted into the ribosome. In the ribosome, the rRNA, not the mRNA, but the rRNA, is a part of the ribosome, and it deals with the next phase of this gene expression, which is called translation. So after the mRNA fits into the rRNA, there is another unit called the anticodon, which attaches on. So basically, there is another small unit called transfer RNA or tRNA, and these bind to amino acids, which are the building blocks of proteins. Also, the tRNA has 3 bases pairs on the bottom of it, which will line up with 3 complementary base pairs on the mRNA. And the base pairs on the tRNA are called anticodons, while the base pairs that it lines up with on the mRNA are called codons. So let's just review really quick. So the mRNA will exit the nucleus, go to the ribosome, be inserted into the part of the ribosome called the rRNA, and then the tRNA are little units that have anticodons which will match up to a pair on the mRNA, which is inserted into the rRNA. So when the rRNA is inside, the tRNA that is bound to an amino acid and has the right anticodon that matches the mRNA It will then bind to the mRNA, and then the amino acid that is attached to the tRNA will be released. And this process will continue until all the tRNAs have bound with the right mRNA and the protein chain is completed as the amino acids of the tRNAs that are being bound are released and they are forming a chain. Also, in regards to the structure of the ribosome and how this task is performed with efficiency, there are certain sites on the ribosome that keep this process actually being able to run fluidly and keep the process going. So we already talked about where the mRNA goes through, the rRNA, but there is also a place called the P site. This holds the growing protein chain, or the chain where all the amino acids are being released and connecting together. There is also an A site. This holds the tRNA that is carrying the next amino acid for the chain. So imagine that the tRNA is bound to the 3 complementary strand codons of the mRNA, and now there's another tRNA getting ready for the next 3 that are coming. And then once it'll keep going in this process and until the strand is complete. Next, we have the E site. This is where the tRNAs leave once they have been released their amino acid. So basically, the tRNA comes in to the A site. It then attaches, it releases its amino acid, which goes to the P site, and then exits through the E site. And this process keeps going on and on and on. And once the strand is completed, a water molecule will attach instead of an amino acid, and this will break the strand away. So this will make sure that the polypeptide protein chain that was created with all the amino acids will be completed because, again, a water molecule will attach instead of an amino acid, and this will break the chain away, and the protein will be completed. So just like how I talked about in the last video about how mutations and things can go wrong when DNA is replicated, there are also things that can go wrong when translation is occurring with amino acids. First, there is the silent mutation. This is a mutation that occurs but has no observable effect on the phenotype. It has no effect on the phenotype because with codons, multiple codon sequences can make the same amino acid. So, for instance, let's just say AUA can make the same amino acid sequence, or can make the same amino acid as AU. So this allows, there's not, even though that one nucleotide changes, it still makes the same amino acid. So this does not make a difference. Also keep in mind that while many sequences can code for the same amino acid, one sequence, take AAG for instance, cannot code for many amino acids. So one sequence can't code for many different amino acids, but different sequences can code for the same amino acid. Next is miscellaneous mutations. These are mutations where one amino acid is changed into another, like substitution mutations of DNA. And these have little effect of the phenotype. The third mutation is nonsense mutation. This is when a point mutation causes the codon to turn into a stop codon. A stop codon is a codon that releases the water molecule to terminate the chain production. This leads to a non-functional protein, as the protein was only partially completed. So I know that was a lot of information, a lot of different processes, a lot of words that sound the same, like transcription, translation, the different kinds of RNA, such as tRNA, rRNA, or mRNA. But this is really the bulk of genetics, and actually how the proteins get created and are used to create your phenotypes based on the genes and gene instruction. And I hope you learned a lot about this process and how the all comes into play and works together. So these are a couple of citations I used for the pictures of this video, and I really hope you liked this video again and learned a bunch. And subscribe to this channel and like this video if you like this kind of video, and feel free to watch previous videos, as this is a series of genetics videos, and there are definitely more to come. And check out some other videos we have on this channel, such as anatomy, book reviews, computer series, and yeah, so subscribe to this channel and leave comments about any questions or ideas you have for future videos. So here's a couple of more citations I used for the pictures for this video. And here the last bit. Here the last bit of citations I used for the pictures for this video. Again, like this video, subscribe, and comment below about questions, ideas, or anything else that you might want me to post about any academic topics. Doesn't have to be genetics or biology. Thanks for watching. Bye.